today's lecture, let us look at biological networks and how network theory is applied to biology, and a very important aspect of this is something known as the centrality lethality hypothesis, which we will discuss in detail, and we will also look at the concept of assortativity in networks and whether biological networks are assortative or disassortative. So, this is a very popular picture that you tend to see in most biology textbooks or systems biology textbooks and so on. This is a classic experiment. This picture actually shows you the results of a classic set of biological experiments which test the essentiality of every node, every gene in yeast. Right. So, this is the yeast protein interaction network, experimentally derived, and every node has a particular color. So, if a node is colored red, it means the removal of that protein from yeast causes the yeast to die. So, it is a lethal deletion. If a node is colored green, it is non-lethal. If a node is colored orange, it is a slow grower, and if a node is colored yellow, we do not have experiments that tell you what happens when you delete the node. Right. So, from this, do you already observe something? Is there anything that is sort of apparent from the picture? Hubs are green or hubs are red? So, many hubs are actually red, right, and some hubs could be green and so on, but you find that majority of the hubs are actually red, and if you see all the peripheral nodes, you will find that they are all mostly green, right, the nodes which have very few connections. So, what you observe, so in fact if you see this network actually contains 78% of all proteins in yeast. So, this is the giant strong component I was talking to you about earlier, right. So, practically all the nodes, 80% of the nodes fall into a single connected component, and although proteins with less than 5 links constitute about 93% of the proteins, only 20% of them were actually essential. On the other hand, there are very few proteins which have a large number of links, more than 15 or so on, like just 0.7%, but nearly 62% of these are lethal. So, the red nodes, so these are all red and these are all green. So, what this tells you is that centrality is correlated with lethality, and this is actually called the lethality centrality hypothesis, very important hypothesis. This is in biological networks because the notion of lethality would not arise elsewhere. So, in biological networks you try to prioritize proteins for lethality or try to rank proteins based on their centrality, and these are the proteins that you want to test for lethality. So, if you want to look at a pathogenic organism's network and you want to try to knock it out, right, you want to kill a disease causing organism, you may want to find the proteins that are most highly connected in the organism and try to knock them out. There are other things you have to worry about, like that should not have a homologue in human and so on, but this is essentially a strategy wherein you find the most central protein in the pathogenic organism and try to knock it out. Yeah, so this has been, so we have actually studied across organisms. So, there is a paper, I probably have it a couple of slides down the line, and there is also this very interesting concept called assortativity. So, let us just quickly look at the concept. So let us look at what is assortativity. So, networks can be assortative or disassortative. Assortative is like nodes connect to like nodes. Here unlike nodes connect to one another. So, what is assortativity? When like nodes connect to like nodes, we say that the graph is assortative. When nodes of different types connect to one another, you can call the graph disassortative, right. As an extreme example, a bipartite graph is sort of perfectly disassortative, right. But if you take any general graph something like this, so in this graph you have nodes of two types, you have red nodes and orange nodes, right, and you see that predominantly nodes of different types connect to one another. You do have one red node connecting red node. So, this is not a bipartite graph, but this is something that you would call more or less disassociative. How do you study or how do you quantify disassociativity or associativity? So, there are two types of measures that you can have. A, if your measure is continuous, and B, if your measure is discrete. So, if it is discrete, you may have something like essentiality, function, or any other kind of property. Continuous, you could have degree or other centrality measures and so on. So, for a discrete case, Newman has proposed a formula which goes something like R equals trace of E, this is a matrix, minus I will tell you what this symbol means. So, this means sum of all the elements in the matrix. And what is this matrix in the first place? It is a matrix structured like this. So, let us say this is essential, non-essential and the unknown, 3 classes, right. This is essential, non-essential, unknown. You will have some value here, some value here, so on. This is your E matrix, right, and this is the trace of the matrix, which is essentially. So, by just counting in the network, I can count how many edges go from E to E, how many edges go from E to NE, E to U and so on. You know, if you go back to the previous graph that we had, the protein network, how many are green to green, green to red, red to yellow and so on, right. We had 4 classes there, slow as well. So, maybe that is a more interesting class to have here. So, essential, non-essential, slow. Fraction. What is the trace of a matrix? Sum of all the diagonal elements.
Sir, if we want to find the disassociativity, why can't we just calculate the trace and divide by total number of fringes?
So, this happens to be a better way to quantify it. So, this number actually goes from -1 to +1. So, -1 would be perfectly disassociative, +1 would be perfectly associative, and values in between are neither associative nor disassociative. So, this is the discrete case. What happens in the continuous case? Would you be able to apply this? Well, yes, you make a couple of bins, make it discrete and apply this, right. So, you say anything greater than 20 links is high degree, anything less than 5 links is low degree, other things are intermediate degree. So, high, low, intermediate, you make 3 classes, make this matrix and you can do some calculations like this. The alternative would be to actually try and plot a graph, right. So, here right. So, let us take an arbitrary graph here. For this graph, for every node you can compute the property of interest, or for every edge, right. So, for every ij belonging to the list of edges in the graph, can you plot the properties? Let us say the property we want is some omega. So, you put omega I here and omega j here, right. So, you essentially plot some statistic. It could be degree, it could be anything else of it could be betweeness centrality. Two nodes of high betweeness centrality connect to nodes of high betweeness centrality.
Sir, one statistic can give.
And you can essentially make a plot of this sort. So, if you end up with a plot that looks like this, it is going to be assortative. If you end up with a plot that looks like this, it is going to be disassortative. Does that make sense? You can compute some correlation coefficient, a Spearman correlation coefficient or a Pearson correlation coefficient to see what kind of correlation is there between. So, that network is assortative on degree, but disassortative on betweenness, right. If one statistic, one omega gives you assortative behavior and the other omega gives you disassortative behavior, fair enough, right. So, you will find that biological networks are typically disassortative when it comes to essentiality, disassortative when it comes to degree, actually heavily disassortative. You may not find much assortativity in biological networks. There is a very good paper called Mixing Patterns in Networks by Mark Newman. This is where he describes these the formula for assortativity that I showed you on the previous slide and so on. For example, especially disassortativity in degree does talk about robustness. High degree nodes do not just connect to one another, right. So, then you remove one high degree node, other high degree node also loses a counterpart and so on, and but it is very difficult to make such sweeping statements about these networks, because in practice these are all global properties, not local properties, right. So, you will have counter examples everywhere. So, for example, you will find a number of essential proteins that have low degree and a number of high degree nodes that are non-essential and so on, right, just to give an example for the a counter example for the centrality lethality. But in general you see that these properties are by and large followed. So, I will also post a reference to our paper where we studied this across 20 different organisms, right, and we find that both betweenness centrality and degree seem to give you a good amount of correlation with lethality. Higher the betweenness centrality or higher the degree, higher the chance of the protein being essential. Shreya, you had a question.
Omega both access the same property values of rocket, right?
Yes. So, degree of I, so let us say, for example, let us say this is degree of I and
An example of omega is degree.
one example for omega would be degree, but you can have betweenness, you can have any other interesting property. In this video, I hope you got an interesting insight into how biological networks are studied using tools from graph theory, and we looked at the centrality lethality hypothesis and also the concept of assortativity. In the next video, we will look at a very important aspect of systems biology, as I always mention, which is what happens to a network when you start perturbing it and how do you study perturbations.